thank you to the organizers for giving me a chance to present some of this stuff. And I'm not just going to talk about this, but I'm actually also going to disprove Nick and tell you that the mouse is not a very good model for the horse. But we'll get to that. So the Marialovics and you started out a number of years ago some trials in adult horses, checking the efficacy of a killed Japanese encephalitis virus vaccine in horses, and in order to fulfill really this about the cross-reactivity. So Australia has got a number of arboviruses that belong in the same group as Japanese encephalitis virus. Japanese encephalitis, we don't have Japanese encephalitis virus circulating in Australia. We've had the odd incursion when mosquitoes blow in from the north, but it's not established here. So the Japanese encephalitis, as many of you will know, belong to the Flaviviridae family and is the type strain for the Japanese encephalitis antigen complex. But we have a number of, does this actually work? Sorry.
So that's the pointer,
okay.
And you can go forward and back with that.
Thanks. So we do have a number of other viruses in Australia that belong to this. Kundian virus is really just a strain of West Nile virus, but much more attenuated. And then we have a rather virulent virus, Murray Valley encephalitis. So one of the aims, just as Nick hinted at earlier this morning, it's just about trying to find cross-reactive vaccines that can provide cross-reactive protection. And that becomes in particular important in the veterinary field where the money really are short, unless you work in cattle, as Tim does. But the horse industry is not, the equine industry is not nearly as generous, despite the fact that they do generate a fair amount of money. So just to give you an idea, we have had incursions of Japanese encephalitis virus into to the Cape York Peninsula a few times, but it is endemic in parts of Asia and Southeast Asia, and then sporadic in a lot of other countries in between. And any horse that goes for competition in any of these countries, Japan, Singapore and Hong Kong and other places, they must be vaccinated. So there is actually a market in Australia for an equine Japanese encephalitis virus vaccine. Such a one doesn't exist. Now, in terms of what does Japanese encephalitis virus do, it's mosquito bird cycle, and then with pigs being a bridge host. And there is therefore also a very good opportunity for the virus to eventually become established in Australia, because we have about 20 million feral pigs up in the northern part of the country. So in one of these incursions, it could establish, and it could start being a major problem for the equine industry as well. What does it do in horses and donkeys? They do develop clinical encephalitis, and it's fatal in most cases. And it gives a really nasty lymphoplasmacytic encephalitis. Now, as I said, Japanese encephalitis is not a current problem, but in a few years ago, we nevertheless experienced a very large outbreak of equine encephalitis in Australia. It was very widespread, especially south of the Queensland border, and in particular in New South Wales, west of the Great Dividing Range, and then even as far close in as Sydney. And it all started in early January after we had had a major flood events, not in the areas that were affected, but in other parts of the country, and then it peaked sort of around April, May, and then it started dying out again. And these were some of the worst affected areas in New South Wales. It did also spread into Victoria and South Australia. Most of the cases in New South Wales were then discovered to be caused by a new strain of West Nile virus that, because it was isolated from a horse in New South Wales, it got the name New South Wales 2011, which I sort of probably will use just for short. It was isolated out of the brain of a fatal equine case, and it was shown to be very closely related to the West Nile virus strain that circulates in North America and has caused a lot of havoc there, both in humans and in horses. However, we didn't see any human cases, and that was probably a little bit of a shame, because if we had, we might have been able to raise some more money for this kind of research. It didn't happen, and the other characteristic of this epidemic was that we didn't see any birds drop out of the sky like they do in North America with the New York 99 strain, where especially corvids and raptors are very susceptible. That doesn't happen here. Possibly because the Australian bird fauna has evolved with lots of arboviruses over time and are not susceptible. It did involve more than 1,000 horses, and the majority of which did survive, but the fatal cases, and I should add that is not necessarily that they died directly from it, but were put down because of severe clinical symptoms, it amounted to about 10 to 15%. Despite the fact that the virus still circulates in the affected areas, we haven't seen this kind of event since then, and that is also so much the more surprising, because we now know that from serological surveys that there's not all that much immunity out there. So that's not explaining. There are a lot of other, just like with diseases in cattle, there must be some multifactorial aspects to these outbreaks. As I said, we've conducted a number of serological surveys, and there's a lot of immunity to Australian flaviviruses that's acquired over the lifespan of a horse. But there's actually very little immunity out there to Murray Valley and congenital West Nile virus. The most common one is to a virus that's called Kookaburra, and it's not in the JEV serocomplex, and apparently does not give cross protection. So there is still the possibility, under the right circumstances, we've got the virus, we've got a large susceptible population, that we could experience another outbreak, and that then also spurred us on to, so just to recapitulate what we wanted to not just conduct these safety and immunogenicity trials in pregnant mares and the young foals for regulatory approval of the Japanese encephalitis virus, but we really also wanted to see if we could get good cross protection in these particular age groups to MVE and to congenital virus. So I'm not going to. Nick has already talked about the adjuvant, but it was we were using Advax, which, as I said, had previously been shown to be acceptable in adult horses at least. And we used an inactivated virocell-grown Japanese encephalitis virus vaccine produced in Japan from the Beijing 1 strain of the virus. We conducted 3 trials. We first had a vaccine trial in foals that were born to naive mares, so they had no antibodies to any of the flaviviruses of the Japanese encephalitis virus complex, and they did, for that matter, also not have any antibodies to any of the other flaviviruses. And then we conducted the second one in pregnant mares, where we vaccinated in the second trimester. And finally, the foals born to these mares were used in a third trial. So in that first trial, we had 19 foals, and we don't sort of coordinate horses in terms of birth. So we had them at ages between 74 and 152 days at age of the start of the trial. 12 of these animals received the vaccine with adjuvant, and then 7 received the Advax only. They were then also, because we had them in open paddocks in an area outside of Brisbane called the Lockyer Valley, and we do know that Murray Valley encephalitis virus circulates out there every so often. So these 7 foals here also kind of functioned as an environmental control for whatever might be circulating at the time. This was at sort of in the spring, in the Australian spring, so mosquito activity wasn't all that great at the time. We gave them an initial vaccination with 12 micrograms of the antigen and 10 milligrams of Advax, and then they were boosted 4 weeks later, and they were bled on each occasion of these vaccinations. And I'm showing you the individual titers from these animals so that you can get an impression of the spread. I should say we tested all the sera initially in a blocking ELISA, but that blocking ELISA just to exclude the negatives, and then we tested any horse or foal in this case that was positive in the blocking ELISA. We tested them by neutralization, and they were slow at seroconverting, but again, perhaps that's because of the age. But we eventually did get pretty good, decent seroconversion to JEV. Now, unfortunately, the cross protection, so in contrast to Nix mice, we were just not seeing the same level of cross neutralization to MVE and to Kundian virus in these horses. We had the opportunity to follow them out to just a bit short of a year, so to 10 months. And again, for Japanese encephalitis virus, specific antibodies, most of them had retained a titer, and I'll get back to that in the last trial. But most of them had dropped out in terms of the cross reactivity. We then went back to the mares, and we vaccinated those in the second trimester of pregnancy, with 11 receiving the vaccine plus Advax, and then we had 4 that just got the Advax, and then we had 2 horses that didn't get either of these ones. The vaccine doses and schedules were the same as for the foals. We bled them at vaccinations and then at foaling, and regularly up until just short of a year. And we also collected colostrum at foaling, and I'll get back to that. So it took a while for them to take. We had most of the mares had seroconverted 4 weeks after the booster, and they retained the titer at least out to 7.5 months, or out to 11 months for most of them. But just as we had seen with the foals, we didn't get the greatest of cross reactivities to MVE and to West Nile virus, which was kind of disappointing. But we at least got good responses to Japanese encephalitis. Now, as I mentioned, we bled the foals at birth and then 12 hours after colostrum uptake. I should say we had some very brave students that was sleeping with the horses literally. They like to foal during the nights, So we had a bunch of volunteer students that were out there in their sleeping bags next to the foals and could collect both blood and colostrum at those time points. So after that, and I'll get back to this about their responses. After that, we used those foals to then vaccinate. After they had received colostrum, again, we vaccinated them when they were around 35 to 73 days, boosted 4 weeks later, and 5 foals born to the unvaccinated mares were given Advax only. Now these are the ELISA results. So all of them were negative. They had an intact placenta. In the horse, immunoglobulins do not pass over the placenta. So they were negative. But as you can see, they all got passive immunity from the uptake of colostrum, and that those antibody titers, now these are ELISA titers, not neutralizing titers, had only subsided in some of them at the time when we vaccinated and then boosted. And in this case, we saw that the uptake was actually somewhat slower than when we had vaccinated naive foals, so some level of inhibition of the immune response. And that was even more evident when we looked at their neutralizing antibody responses. We didn't, despite the fact that all of them did have a response in the ELISA test, we didn't get neutralizing antibodies in all of them, and the neutralizing activity was really very, very short-lived. So just to put this into context, so as I said, after suckling we had all of them. We didn't get colostrum from one of them, or blood from one of them, but they all had tremendous got good passive immunity, and some of that, despite the fact that some of these mares didn't really have very good cross-reactive responses in terms of their serum antibodies, most of them had quite strong cross-reactive activity in the colostrum. So maybe colostrum is a good source of that there's other things in that colostrum that helps that neutralization activity. We did get a slow response to this, but as you will see over time, that 9 months out, they had all dropped off. We didn't have a single animal that had any neutralizing activity left, and that was a real concern of ours. That not a surprise, because we all know that passive immunity in young animals is going to inhibit a vaccine response. So it wasn't a surprise as such. It was nevertheless a disappointment that despite Advax adjuvant, that we didn't see a better response. However, when we then went around, and so this is the bottom line in my previous slides, where they're all negative at that stage. And then we boosted them, just one boost, and within 2 weeks they had all seroconverted with really high antibody levels. So despite the fact that initial vaccination in the face of passive immunity had not had a very good response at the surface of it, we had obviously managed to induce a really good memory response, because this was just a single vaccination with 6 micrograms of antigen, and they all seroconverted within 2 weeks. And in this case, the antibody response also lasted longer. We even got cross-reactivity to MVE in this, but unfortunately not to West Nile virus. So To conclude on this, the inactivated Japanese encephalitis virus vaccine is safe in foals and pregnant mares, which is, one step to getting it registered here in Australia. It provides protective immunity to Japanese encephalitis up to at least 10 to 11 months. The cross-protective immunity to MVE and Kundian is unfortunately more stochastic, but does seem to be related to the initial antibody titers against Japanese encephalitis. And then, as I said, no big surprises. Passive acquired immunity will interfere with the quality of subsequent vaccine responses. However, at least in this case, we got very, very good memory responses. And that is also worth keeping in mind, that even if you don't go out and you don't have the opportunity to revaccinate, it would mean that should they encounter the virus in a natural infection, they probably are still protected even when we can't measure their antibody responses at that stage. And I owe the horses a lot. Worked with a bunch of very wonderful mares and foals, and I could not have done this without Mitch Coylan and Lisa Duma, who are horse whisperers. And then on the laboratory side, some of my students and colleagues from the School of Veterinary Science at the University of Queensland, and in terms of the serology, lots of members of the Roy Hall lab. And Roy Hall will talk more about some of these vaccines again tomorrow, some of the arbovirus virus vaccines that we really do need. And a big thank to Nick for providing us with the vaccine and encouragement and other funding, and the rest of our funding we got from UCU CIF and ARC Linkage Grant. Thank you very much.
So I'll open for questions, but I must say, in terms of the mice lying, in fact the mice didn't lie, because for instance, when we immunised with the Japanese encephalitis with a single dose with the Advax, and then challenged with West Nile, although the animals were completely protected against West Nile, they had no measurable neutralising antibody in their sera prior to challenge. So what we believe, and I guess that's in some of the papers we've published, is that the vaccine's inducing a memory B cell population that's cross-protective. But that doesn't show up at the level necessarily of the serology. But as long as you have those memory B cells, and Mario even transferred the memory B cells and actually showed he could transfer the cross-protection without the sera, just moving the B cells. So maybe, again, in these horses, even when the serology is absent, doesn't mean they're not protected.
And unfortunately, we haven't been in a position to do challenge experiments. We don't have the facilities to do it with Japanese encephalitis, and we haven't until recently had the facilities for doing MVE or Kundian, and now we need the funding to do it, if we were to do it.
Yeah, so I mean, I guess it shows the critical nature of being able to do challenge studies, which in large animals is obviously a hell of a lot more difficult and costly than doing it in small animals. But hopefully, fingers crossed, there may be opportunities to confirm that horses and mice agree when you actually do the challenge. So any other questions? Yes, at the back.
This was the same vaccine that was used in humans, right?
Yeah. Yeah, it's a human vaccine.
Which is formaldehyde fixed. Killed.
Mm-hmm. Yeah.
And what was the dose used in horses the same as humans? What was the...
Yeah, it is the human dose.
You use the human dose?
Yeah.
Did you want to add to that, or?
Well, I'm surprised.
Surprised it did well, or surprised?
Yeah, so you didn't increase it because of the weight?
No, because it also, there's also the aspect to it, Andreas, that it also has to be economically viable. and you're never going to convince a horse owner to invest in more than what it would cost to vaccinate a human. I can guarantee you that.
It's an expensive vaccine.
Yeah, it's about $200 a dose. Well, in Australia, if you buy it in Australia, it's $200. I think in Japan it's about $40 a dose, so it's a lot cheaper. But certainly in Australia, it's about $200 a dose. And it's in fact as an unadjuvanted vaccine, it's a 3-dose vaccine. So that would be $600 for a full course. We've actually, with the adjuvant, reduced it to a 2-dose vaccine. So again, this was a fairly extreme test of the system. We were using a human dose in a much bigger horse, and we're only giving 2 doses, whereas in humans you actually have to give 3 doses to get protection.
Yeah, I mean, it's worth keeping in mind that these foals, although they're young, they at that stage already weigh as much as an adult male or the double. And then the mares are up at 400, 500, 600 kilograms.
Okay, so thanks very much.